All right. Thank you, Elizabeth. We appreciate your help. And Peter, thanks for joining us today. And I also want to welcome all of our participants. Thank you for signing in for this webinar. This webinar is hosted by Cambridge Healthtech Institute, and it's specifically sponsored by Sino Biological. I'm Chief Business Officer for Sino, and I'll be your MC today. So I would like to officially welcome everybody and thank you for the very strong registration and turnout today. The purpose of this webinar is to discuss the latest in vaccine development for COVID-19, but also the development of general research reagents. So with that, I know we're pressed for time. I'd like to introduce our first speaker. It's quite an honor to introduce Dr. Peter Hotez, who will be our first speaker today. Peter is Dean of the National School of Tropical Medicine and also Professor of Pediatrics and Molecular Virology and Microbiology at Baylor College of Medicine. He is also a fellow at Texas A&M, and he holds a variety of fellowships at Texas A&M. And he is an endowed chair of the Tropical Pediatrics Department at Texas Children's Hospital. As head of the Texas Children's Hospital and Center for Vaccine Development, Peter leads a team of researchers that focus on product development partnerships and really developing new vaccines for a variety of neglected viruses. So given this point, it's quite an honor to have Peter as our keynote speaker today. And Peter, at this point, I'll turn it over to you. I want to mention that you've got 15 minutes. You can go a little longer if you like, and we'll have a 5-minute Q&A session after that. And I'll be taking questions from the audience via chat, and then I will relay them to you, Peter. So thank you, and the floor is yours.
Well, thank you very much, and thank you to Sino Biologicals. You've been a tremendous source of reagents, and making really important contributions to our fight against COVID-19. And just want to use this opportunity to thank you and your amazing group of scientists for getting out those reagents so quickly. It's really truly been life-saving, and I appreciate all you've been doing. I think one of the, just in terms of the big picture overview, this is a new ebook or instant book that's coming out. I like to write books. And one of the observations we made a few years ago is that when we think about global health problems, we always think of it as some of the poorest, most devastated countries in the world. But some of our observations are that the biggest global health threats are actually among the poor living amidst wealth in the G20 countries, the G20 economies. And I think COVID-19 is a pretty good example of that because we're seeing COVID-19 increasingly play out as a health disparity in the G20 nations, which overwhelmingly are the nations affected. So the United States, of course, the European countries, Russia, Brazil. These are the places where COVID-19 is striking. But what we've seen in the U.S. especially is the poor, especially people of color, African Americans, Hispanic Americans, and Native Americans particularly affected. So this concept of the poor living among the wealthy is blue marble health, to give it a different name from traditional norms of global health, which look at developing countries versus developed countries, to talk about the poor living among the wealthy, and this will come out next week. And what we're seeing is we're hearing about COVID-19, of course, in the United States and Europe, but now this virus is starting to plow through the big favelas in some of the Brazilian cities like Fortaleza and Belém, and Guayaquil in Mexico. And I think this is going to be a new chapter now of COVID-19 as it becomes such an important global health scourge. And of course, a vaccine is needed. One of the things that I like to say when I talk about vaccines is the technical feat of making a COVID-19 vaccine is actually not going to be that difficult, I think. Carl Zimmer just had a piece in the New York Times where he talked about the SARS-2 coronavirus being a clumsy virus, meaning it's not terribly clever. It doesn't vary. It's antigens that require broadly neutralizing antibodies. We think like HIV, for instance, but in fact, it's a really straightforward problem of creating an immune response against the spike protein, shown on the upper right. Those spikes coming out of the ball there that dock with the host receptor and bind to the angiotensin converting enzyme receptor, which is itself is an enzyme. It's a carboxypeptidase. You create an immune response against the spike protein, especially an antibody response, and especially neutralizing antibodies that prevent the virus from binding to target cells, and you'll have a vaccine. Then it's a question of which of those technologies is going to be the most effective in inducing an immune response and creating neutralizing antibodies against the spike protein. That is the major technical hurdle. And on the left are a bunch of different vaccines going into clinical development. This is a slide that Anthony Fauci, when he spoke to the National Academy of Sciences a couple of weeks ago, and there are different ways potentially to do this. So a recombinant protein, that's why it says Baylor and collaborators. There are nanoparticle approaches, genetic immunizations, DNA and RNA vaccines. We've been hearing about Moderna and Inovio, adenovirus-based vaccines like the Oxford, now with AstraZeneca, J&J, but they're all fundamentally around creating an immune response to the spike protein. So you've been hearing about more than 150 vaccine candidates. All of those will not go into clinical trials. We're looking at the first round of 7 in the U.S., and those are the BARDA vaccine candidates that will be scaled, manufactured, and hopefully will be in the second round of 7 that'll go into clinical trials later in the fall. And then there's about 4 or 5 candidates coming out of China and several in Europe. And I'm fairly optimistic that several of those will actually turn into bona fide vaccines that goes through phase 3 trials. And in the US, those phase 3 trials will start in the summer, and I think beginning in July with the first Moderna vaccine, and they'll continue over the course of a year. By the third or fourth quarter of 2021, we may have some vaccines that potentially could be released to the public. Our particular vaccine is being done as a partnership with PATH, which used to stand for the Program for Appropriate Technology in Health. And this is an organization that led the development of the meningococcal A vaccine for Africa, the malaria vaccine for Africa, and we're partnering with them because we think ours, which is a recombinant protein vaccine, is going to be a low-cost vaccine that'll be affordable and accessible and uses the same technology that's used to make the recombinant hepatitis B vaccine that's made in India and Brazil. And PATH has that bandwidth to be able to advance vaccines through global health, and we're hoping that that's going to be a possibility. So our center is based in Texas at the National School of Tropical Medicine, the Baylor College of Medicine, and it's within the labs of Texas Children's Hospital. That's a picture of Dr. Mary Elena Bottazzi on the lower left, my science partner of 20 years, and we work together to develop the vaccines, and we use a low-cost recombinant protein technology for global health vaccines. And here's our portfolio on the right, which was mostly around neglected parasitic infections. And then about a decade ago, we took on a coronavirus vaccine programs because for a decade, and indeed up until the end of last year, nobody cared about coronavirus vaccines. So even though we were not virologists, coronavirus vaccines had been orphaned and needed somebody to take them on. And that's what we've been doing for the last decade. So ours is a recombinant protein vaccine that encodes the receptor binding domain of the spike protein. Here are some of its specs. It's produced in yeast. What's great about it's being produced in high yield and at low cost. And it's actually made from the first coronavirus that has about 80% similarity between SARS-1 and SARS-2. And we've just put up our data on bioRxiv on its, we've been publishing about how we're scaling up production, which is several papers out on that. And we have now under review, and it's up in bioRxiv, a paper showing getting high levels of neutralizing the antibody and 100% protection in transgenic mice with the ACE2 receptor. Another aspect of what we do is to avoid some of the immunopathology that's seen with experimental coronavirus vaccines. And you hear a lot about ADE, antibody-dependent enhancement. We see it a little bit differently. You know, from our decade of working with coronavirus vaccines, we actually see two phenomena depending on the vaccine that you use. One is a cellular infiltrate in the lungs and liver that you could see upon virus challenge. We call that eosinophilic immune enhancement. We see it more with some of the virus vaccines. And we actually find that if you administer a vaccine on alum, it greatly reduces the eosinophilic immune enhancement. And that may be counterintuitive to some because they think it's due to a Th2 response. We find it's not. It's really a Th17 response with high levels of IL-6, and that the eosinophils come in just like they do with inflammatory bowel disease or asthma, linked to Th17 responses. There's also an antibody-dependent enhancement that we've seen in vitro with coronavirus, but not really in vivo. So people are drawing parallels to dengue and maybe RSV. We see this potentially as a less of a pressing problem than the eosinophilic immune enhancement. So here's an example of what it looks like. If you immunize laboratory mice, and you can see this in primates as well, if you challenge them with the virus, depending on the vaccine you use, you can create this eosinophilic immune enhancement. So on the lower right, those brown dots are the eosinophils on immunohistochemistry, and you see a lot of those eosinophils using the full spike protein. But we find if we engineer out those epitopes and just focus on the receptor binding domain and use high amounts of alum, you can largely or completely abolish the eosinophilic immune enhancement. So this is our group that we've assembled together now with PATH to develop that program. Lastly, I want to finish by, we're hearing a lot about this Operation Warp Speed language that's coming out of the White House. And that actually gives us pause for concern, because one of the other hats I wear, I'm both a vaccine scientist and I do a lot of advocacy around vaccines because I have a daughter with autism and wrote a book called Vaccines Did Not Cause Rachel's Autism. And I'm very concerned about the fact that some of the language being used, also some of the press releases coming out of the biotech saying we're going to have a vaccine in weeks and months. That's potentially damaging. This is what's happening in Texas right now, where we're up to 64,000 kids not getting access to their vaccines because their parents have stopped vaccinating them because they're concerned that vaccines cause autism. And I've started, I wrote a book about a year ago, a year and a half ago, called Vaccines Did Not Cause Rachel's Autism, to sort of debunk some of the false assertions that vaccines cause autism alleged by the anti-vaccine lobby. And that's a picture of me with Rachel. But now, because of the Operation Warp Speed and the press releases coming out, we're going to have a vaccine in weeks and months. We have to remember that the central tenets of the anti-vaccine movement are, one, that vaccines cause autism, which I've been working to diffuse, but also that vaccines are rushed, that they're not safe, and that there's a cozy relationship between pharma and the U.S. government. And unfortunately, a lot of that language is now fueling into the anti-vaccine movement, to the point now where some surveys are showing that even if the COVID vaccine is available, a significant percentage of Americans will refuse to take it, to the point where we're working now with a group at City University of New York to look at the fact that we may not have enough Americans willing to take the vaccine in order to create herd immunity, which is something I never would have dreamed of a few months ago, but that is the reality now. And so maybe we can talk about that at some of the Q&A. So I'll stop there and see if, I don't know how you want to proceed, whether you want to take a few questions or really up to you.
Thank you very much, Dr. Hotez. I appreciate that. That was a brilliant talk, and I'm sure, as well as many others, look forward to reading your book about vaccines and how they do not cause autism. So thank you once again, and I'm going to take questions now from the audience, and I'm already seeing a number of questions popping up here under the chat, so I'll just start with the very first one that we received. One individual is asking, could CRISPR and the Cas9 system be utilized to develop a vaccine? And if so, what are your thoughts on the ethics behind that?
Well, as I say, I think, for this particular vaccine, I think it's relatively straightforward in terms of what has to be done in terms of creating a protective immunogen. It really need to focus on the spike protein, especially the receptor binding domain. Potentially, CRISPR-Cas9 could have an alternative role, which is into helping engineer monoclonal antibodies that are specific for the spike protein. And there, I think there's some important therapeutics that could be made available. We're now so far convalescent plasma patients who've recovered and have sufficient titers of neutralizing antibody. That could be harvested, and so far it's been given to about 4,000 patients in the United States with COVID-19, and the Mayo Clinic is now doing a big data analysis to look at the effects. I think that's going to be very promising. The problem is it's resource limited because it requires having patients around with COVID-19. So the next generation is going to be making hyperimmune globulins or making monoclonal antibodies that are specific for the S protein to prevent it docking with the ACE2 receptor. And I think when that docks with the ACE2 receptor, I think that's when you start seeing a lot of the coagulopathies that we're seeing, the pulmonary emboli, maybe thrombosis in the coronary artery, and strokes. So having those monoclonals, I think, will be very important, provided they're given early enough in the course of infection, and also developing this as a form of post-exposure prophylaxis, like we're seeing in the White House. Maybe one day, if we think the president has had a significant exposure, we'll have one of those monoclonal antibodies.
Great. Thank you for that. And for the sake of time, I'm going to ask you one more question that a number of folks are chatting about online, and I think this really hits home. Is once the vaccine is successfully developed, what are your thoughts regarding it being available globally? And the reason I ask this is, companies like Moderna need to make a profit, but individuals are also going to need to be vaccinated for a very reasonable cost. So how do you balance that?
Yeah, no, it's a great question. And that's why, we've been taking a different approach since our whole business plan, if you call it that, is around low-cost, affordable global health vaccines. So you're absolutely right. I mean, if the Moderna vaccine and others are proven to be successful, we don't know at this point, because it's a brand new technology, what the scalability is going to be, and would it be made available to Brazil and India and sub-Saharan Africa. That's why we're moving ahead with a low-cost recombinant global health vaccine for COVID-19, initially with the SARS-1 and then moving to the SARS-2 as well. And because we know there is that scalability there. So we think, I think it's the other point a couple of points I'd like to make relative to that. It's not like there's going to be one COVID-19 vaccine. I think there could be 4 or 5 or even more. And between those, hopefully we'll be able to cover the global population, maybe including ours. Also, I should point out, if you look at the history of vaccines, usually the first vaccines that are released are not necessarily the ones that have the staying power. So we saw this with rotavirus. The first rotavirus vaccine was not on the market very long. It was taken down and replaced by 2 others. Same with the HPV vaccine. So they get improved upon. And this has been the history of vaccinology, the Haemophilus influenza type B vaccine. So likely the COVID-19 vaccine. That's why I tell, my partners, be careful what you wish for winning the race to the COVID-19 vaccine, because history says the first may be not around very long, and it's the ones that come afterwards that may be around the longest.
Yeah, very good point, Dr. Hotez. Thank you. We've got a lot of other questions, but for the sake of time, Elizabeth has agreed to download these from the chat room and she will send them out. And if you could just quickly answer the ones that you prefer to, we would really appreciate that.
Okay, well, thank you again.
Thank you. So now moving on to our next speaker, who is not in attendance today. That is my colleague and good friend, Dr. Chia-Hui Yang. And she is the Director of Research for Sino Biological based in Beijing. And she is an expert at recombinant protein development and antibody development. Jia Wei received her PhD from Tufts University, and she joined Sino 10 years ago. And like I said, is currently the Director of Research and Development there. In her capacity there, leading our R&D team, she and her team has developed greater than 3,000 recombinant proteins and greater than 5,000 monoclonal and polyclonal antibodies. And I can say with great pride that Chia Wei also has led the development of the world's most comprehensive selection of COVID-19 related research products and reagents available from Sino Biological. So since it's about 3 a.m. in Beijing now, she unfortunately was unable to attend, but she did record her presentation. And my colleague Elizabeth at CHI is going to help us to present that now.
So good afternoon. This is Jiahui Yang from Sino Biological. So today I would like to talk about some strategies from our companies for making COVID-19 reagents to support SARS-CoV-2 research, diagnosis, and vaccine development. And for this talk, we mainly focus on the antibody development. We will share the information about the decisions and the challenges we made during the development processes. So this talk will divide it into 5 major parts. First, we'll just briefly go through the SARS-CoV-2 and what is this virus is and what is the key factors about these viruses. Then we will talk about the strategies for making SARS-CoV-2 antibody development, and then we will go through some results and the challenges we have made during the antibody development. For the last 2 parts of the talk, we will just share some other related tools for the SARS-CoV-2 research and another PRO-VIR, our viral protein fraction. As we all know, WHO has characterized the COVID-19 as a pandemic, and these viruses has caused a lot of public health threatening and also affect our economic development a lot. So to study COVID-19, there's a lot of urgent need for the research tools to help the research and diagnosis and therapeutics. This slide is just showing the complete genome of the SARS-CoV-2. This virus has 4 major structure proteins, M, E, S, and N, 14 non-structured proteins, and other several accessory proteins. So our work is mainly focusing on spike and the N proteins, just as other people did. This is just a diagram showing that what this virus looks like. So we can see that the S protein is on the surface of the virus, and this is a very key protein for the viral entry. It helps to interact with the ACE2 protein on the human cells and help the virus infecting the human cells. Therefore, spike is the primary target for therapeutic antibodies and diagnosis. And for the recently developing vaccines, most of them are targeting spike proteins. And for this protein, I want to add a few more informations. The spike protein can be divided into S1 and S2. There's also a very important domain on the S1, which is the RBD domain. This is the key domain for interacting with the ACE2 protein. The other key molecules we will talk about a lot in the following slides is the N proteins. These proteins is a very sensitive marker when people study SARS, the original SARS viruses. So when we compare the sequence alignment for 7 known human coronaviruses, we found that for the S proteins, the SARS-CoV-2 and the SARS has a very high identity, which is similar for the N protein. So based on this information, we did the following strategies for making the reagents for both antigen and antibodies of SARS-CoV-2. Because there's a very high homology sequence alignment between SARS and SARS-CoV-2, we try to do the first strategies by testing the original SARS antibodies and the SARS antibody libraries to see if we can find some antibodies and recognize the SARS-CoV-2. Because the development speed is very important at this time, if we can obtain the good antibodies or other reagents for supporting the SARS-CoV-2 scientific research, it will help a lot for the following vaccine development and therapeutic development. And the third strategy is start from the beginning. We will immunize the SARS-CoV-2 antigen and then do the antibody development. But the first key reagent we need to have is the antigen from SARS-CoV-2. We will use recombinant technologies for developing the recombinant SARS-CoV-2 antigens. Here are the 4 major expression hosts available now for recombinant protein expression. And when we go through the information we have about the SARS-CoV-2 spike protein and N protein, we know that the spike protein is highly glycosylated. So here is just a diagram from one reference showing that there are a couple molecules can be modified on the RBD region. We also see that in our results when we using HEK293 cells and the insect cells for expressing the part of the spike protein. We see the molecular weight differences in these 2 systems because of the glycosylation pattern differences. Therefore, we chose the eukaryotic expression host, HEK293 cells, and the insect cells for expressing the following S and N proteins. So here are several proteins we obtained for after the recombinant expression. We produce the RBD and SECD S1 and several spike fragments. We also successfully obtained the N proteins. When we check their HPLC properties, we can see a high percentage of the one spike component for the proteins. Then we are coming to our strategy number 1 and number 2. We screen the SARS antibodies or the SARS antibody libraries for SARS-CoV-2 antibodies. This is one of the antibodies we obtained from the SARS spike antibodies. This is the rabbit monoclonal antibodies. When we check this antibody, the interaction between the antibody and the SARS-CoV-2 spike proteins, we can see their interaction very well. And then we also retest these antibodies to see whether can recognize the pseudoviruses of the SARS-CoV-2. One, the pseudoviruses infect the ACE2 expression HDK293 cells. We can clearly see the interaction between the antibody and the pseudoviruses. The bottom part of the EM picture is from one of the references, which is citing our products. And this monoclonal antibody products, you can see we got very good EM results, showing that we have successfully obtained one spike antibody, which can recognize the SARS-CoV-2 spike. We got pretty much the same results for the N proteins. We rescreened the SARS and N antibody and the N antibody libraries and obtained one rapid monoclonal antibodies. And these antibodies can recognize the SARS-CoV-2 N protein and also can work for the Western blot applications. Our customers use these antibodies for IHC analysis. We got very good pictures to detect the NP antigen in the kidney tissues. Then we want to check the affinity of these new antibodies. And we found that both antibodies show very high affinity interaction with their proteins by octet. So for the strategy number 3, we want to start from the beginning. We immunize the animals with the SARS-CoV-2 antigens, including the spike and the N proteins. Since we want to obtain the antibodies which hopefully they can recognize the natural viruses or they can have the neutralization functions. So we chose recombinant proteins as the immunogen. We immunize the immunogen in both mice and rabbits to do the antibody development. For developing platform has been used for the new antibody generation. The regular polyclonal antibody platform, the classical hybridoma. We also use phage display library and B-cell antibody cloning techniques for monoclonal antibody generation. Then lastly, we'll test the application validation by ELISA IHC neutralization. We also want to test if this antibody is specific for SARS-CoV-2 or if it has cross-reactivity to other coronaviruses. So this is just one of the results we obtained for these new antibodies. There are 6 antibodies showing that we are testing the interaction by ELISA with SARS spike proteins or SARS-CoV-2 spike proteins at 2 different concentrations of the antigen. And we see that for the antibody number 1, 2, 3, these 3 antibodies can interact with SARS-CoV-2 spike, but did not interact with SARS, the original SARS spike proteins, which means this is probably the spike-specific antibodies. We didn't show the results here, but we do test these 3 antibodies to see if they can recognize other 5 human coronaviruses, and we found that all 3 antibodies are specific only to the SARS-CoV-2. So we successfully got a very high specific specific antibodies, which only recognizing the new SARS-CoV viruses. Then we did the neutralization assays and found 2 antibodies which can neutralize the interaction between the pseudoviruses and the ACE2 expressing cells. So currently we have a good quality SARS-CoV-2 antigens, and we also obtained several specific SARS-CoV antibodies. So we did a lot of combination assembly, several kits for different areas of the applications. the left chart showing that this is a inhibitor screening ELISA kit. So this kit is used for screening the antibody inhibitors, which is for selecting the neutralization antibodies. We also have the antigen detection ELISA kit by one pair of antibodies, and then we can quantitate the amount of antigen in your sample. We also have the antibody titer and assay kit, which help to detect the immunization titer of the antibody you obtain in the samples. Meanwhile, we are still working on selecting more SARS-CoV-2 antibodies, not just for the specific antibodies. We are also willing to obtain some antibodies which can recognize the SARS-CoV-2 and the other coronaviruses. So the study is still ongoing. To summarize of our talk today, and we think we are in highly demand for high-quality SARS-CoV-2 reagents. And so this is Sino Biological is working hard on this area. For antibody development, no matter for SARS-CoV-2 or other antibody development, we believe antigen with good qualities is a very key factor. We use several strategies for developing the SARS-CoV-2 research tools, especially for the antibody development. And the first strategy and second strategy, we successfully get the SARS-CoV-2 antibodies from the original SARS antibodies or the SARS antibody libraries. Both antibodies show very high affinity for binding to the antigen. Currently, we are still having a lot of challenges for obtaining the broad spectrum antibodies which can recognize all 7 human coronaviruses, but we are working on that. For now, we have already had several collections of the SARS-CoV-2 research tools, including the antigen and antibodies we have covered in this talk. And we also mentioned we have several ELISA kits for detecting no matter the antigen or the antibodies for the research. This is our last slide. We just want to mention that other than SARS-CoV-2 research tools or other coronaviruses research tools, we have a very large collection of viral antigens, including antigens to influenza, HIV, RSV, and also the antibodies to all these types of viruses to support the viral research. And thanks for your time.
All right, Jia Wei, if you were here in live, I would thank you in person for that wonderful presentation. I want to emphasize that I think one of the main points of Jia Wei's talk was that you need a very, very high quality antigen in order to produce highly specific and accurate and high affinity monoclonals and polyclonals. And I do know that at Sino, we generate probably about 85% of all of our proteins and antigens in HEK293 mammalian cells. And the reason for that is we can generate literally gram quantities if needed. But much more importantly, we can generate properly folded and properly post-translationally modified proteins. And as Jia Wei mentioned, with the spike protein, it's heavily glycosylated in vivo. And so you really have to recapitulate that phenotype when you're generating an antigen to produce an antibody, or more importantly, to produce a vaccine. So anybody that has questions for Jia Wei, please type them into the chat box. And what we're going to do is email them over to her, and she will get those questions answered. So I guess with that, moving right along, we'll move on to our last speaker, and last but certainly not least, he's a colleague of mine. His name is Dr. Farshad Girahu, and he is the Chief Scientific Officer for Atlanta-based GeoVax, one of the leading premier companies in vaccine development worldwide. Dr. Jirahu is an expert at developing vaccines, and he has specifically worked with his team to develop the MVA VLP vector technology platform, which is proprietary to GeoVax. So in 2014, I want to mention that Dr. Jirahu was named one of the 50 most influential people worldwide in vaccines and vaccine development by Vaccine Nation. He is the author of numerous patents and over 100 peer-reviewed journal articles. And his patents have actually resulted in a number of commercialized vaccines. Dr. Jirahu obtained his PhD in virology from the University of Vienna, Austria, and also holds a master's in science and genetics. So the title of Dr. Jirahu's talk is Development of a Single-Dose Vaccine Against COVID-19 using the MVA VLP Vector Platform. Dr. Jirahu, it's an honor to have you today, and thank you for presenting.
Thank you very much. Good morning, good afternoon, and good evening for everyone. I think I have about 15 minutes time. I try to make sure that we can cover all slides I have here. Before I go to COVID-19, I give you an overview of the platforms so you make familiar with other indications that we have already developed, and I talk about a little bit of hemorrhagic fever vaccine and then switch to COVID-19 and summarize that. So this is the basic overview of our platform. The platform uses MVA, which brings the safety of the platform because MVA has been used in the last 50 years as a vector and also as a safer smallpox vaccine for immune-compromised people who cannot get the vaccinia or regular smallpox vaccine. So we have the safety coming from the parent MVA, but we made it also immunogenic by delivering antigen in a VLP format. So as you know, the VLP vaccine made in its subunit like HPV and HPV, they are a single antigen and they are highly immunogenic. They can be used in microgram quantity and they are generally provide subtype specific immunity. But we made the VLP of more than one antigen. And the idea is that to broaden the immune response against the strain of the pathogen which might not be present in your vaccine. So here show the construction, the genome. So we put different antigen on the vaccinia promoter. So when you use vaccine in mammalian cells, you inoculate vaccine with your live vaccine, which is infectious dose, just like any other vaccine. But the virus does not produce more MVA, so that brings the safety. But rather, they produce the antigen which has inserted here, and here you see the VLP coming after vaccination of subject in vivo. So the VLP are produced in people. So we use in people factories as a host to produce VLP rather than producing outside and have to purify it and refold and unfold and so on. On the right, you see the Ebola, Sudan, and Marburg and Lassa, the VLP produced in the cells, and they budding from the infected cells, and they just look like the real virus, but these are non-infectious virus-like particles. So here shows the overview what the advantages. I mentioned the inherent safety of the MVA. MVA was licensed actually 2019 by FDA. For prevention of smallpox and monkeypox. Our MVA VLP HIV has been in 6 human clinical trials, and these have been prophylactic trials and more than 500 subjects, excellent safety similar to placebo. And we are planning to run 2 therapeutic clinical trials in this year. So it's non-replicating in the host, and we activate both humoral and cellular arm of the immune response. I think it's very important. And we induce a Th1 biased response. And this is also an important hallmark of a COVID-19 vaccine. And I show some data that we can induce sterile immunity after a single dose in very severe challenge animal model. I show you some data about this highlighted area, Lassa fever, Ebola, Sudan, and Marburg, and Zika, and also coronavirus. Obviously, we use the platform for immuno-oncology, malaria, and therapeutic indication for hepatitis B. So it's a broad platform. Here, so if you focus on the right one, this is the non-human primate experiment with Ebola, or MVA-VLP Ebola. As you see, a single dose prime on the right side or 2 doses prime base made no difference. They have 100% survival non-human primate, whereas the control died. We induced the antibody response on the left side, and the level of antibody response produced after 1 dose was similar to those produced by Ebola, VSV, Merck vaccine, and also adenovirus. After about 10 days post-vaccination, we were able to induce protective antibody. We also induced neutralizing antibody, ADCC, and if you look at viremia in the right corner, these are the control animal. More than 5 log of live virus was detected in the blood after challenge. Absolutely no viremia in single or 2-dose immunization in non-human primate. So this is very unprecedented from a replication deficient vector that producing almost sterile immunity. So we repeated with the Marburg vaccine. This is the guinea pig experiment. Again, we induced broadly neutralizing antibody or binding antibody and neutralizing antibody. And the vaccinated animal post-challenge, absolutely no weight loss. It controls black line, they lose weight and they die. And the survival in the middle, 100% survival in all animal, 100% death in control animal like black and white. If you look at viremia post-challenge, again, under the level of detection on plaque assay, which is 2 log, 10 PFU, absolutely no viremia in vaccinated animal, whereas high viremia up to 7 log in control animal, the black dot. We go to Sudan, we see exactly the same thing we have seen with Marburg and Ebola. Prime boost, prime single dose, prime boost animal, red light on the left top, 100% protection, 100% death in control animal. Weight loss, again, no weight loss in vaccinated and challenge animal, severe weight loss in control, and they die. If you look at the left corner, this is the viremia post-challenge. All the black dots, they are control animals, and the red one is the prime, single dose, and the blue is 2 doses. As you see, the level of detection here is lower because the plaque assay was done with undiluted sera. Generally, you dilute the sera 1 to 5 or 1 to 10. This was completely undiluted sera. As you see, the one single blip on the prime, on the red one, on day 6, and 2 single blip on the prime boost on day 8 and 10. Absolutely everything else below the level of detection. If you look at the right, is the score, health score, absolutely no score on single dose or 2-dose vaccinated animal. We move to Lhasa, exactly the same. Single dose, 100% protection. We move to NS1, Zika vaccine, again, 100% protection. In non-human primate, despite NS1 antigen, which produced a non-neutralizing response, we had also control of viremia, significantly control compared to the control animal. And we also induce nice seeded T-cell response with multi-cytokine production. Obviously, if you want to use a vaccine for one indication, you want to be able to give the same vector having another vaccine. In this case, we show that if we go with the MVA-NS1 for Zika, vaccinate, we can go back to the same animal and give them a Ebola vaccine, and absolutely no vector immunity and no interference in induction of the anti-GP antibody. So we switch to COVID-19. Here you see the picture of variant of the coronavirus COVID-2. There are basically 4 structural protein on the surface. The major spike protein, yellow, and there is also M protein, matrix protein, and also an envelope protein, is a pentamer. So you see the next slide. what we have in our first two construct is everything you see on the surface: spike protein, matrix protein, and E protein. And this is basically our vaccine candidate, which is supposed to make the VLP. We did not include the N protein because with SARS-1, N protein was shown not inducing any neutralizing antibody and also caused immunopathology with SARS-1, obviously. So this is an important slide. On the left, this is GeoCMO1, CMO2, and CMO3. So the first one is using S antigen, envelope, and M antigen. As you see, the structure of S protein is a natural structure, which many companies chose that natural structure in the construct, that including the Oxford group using adeno, chimp adeno, Johnson & Johnson using AD26, and also CanSino using AD5 vector. They all use the natural spike protein structure. There are other companies like Moderna, mRNA, and also Novavax with this subunit S. They use the pre-fusion stabilized structure, which is used in our second construct. So also novelty of our construct is that we are using more than one S protein, and we use the pentameric E protein, as you saw in previous slide, and we also M protein. And we were surprised that there is a paper coming in Cell. It's a pre-proof. It's not published, but pre-proof is available online. Surprisingly, these authors showed that there is a predominant response in subjects which did not hospitalize by coronavirus infection, COVID-19, but they had a mild disease. So when they looked at the MHC class 1, MHC class 2, CD4, and CD8, using a mega pool of peptide, they screened the whole genome response in this patient, and they were surprised that they saw the very dominant portion of CD4 T cell response and also CD8 T cell response were directed to the M protein. And this was not expected because M protein did not play a major role in terms of providing T cell epitope in other SARS coronaviruses. So as you see the circle on the right panel, this is the CD4 to the S protein, which is dominant, but also the green line, that this is the M protein CD4 response. And also on CD8 T cell response, it's proportionate to S protein and M protein. And S protein in this case is not dominant CD8 T cell response, but it's also balanced with the M protein. So we are pleased that we have the M protein in this construct. Obviously, we need it for VLP formation. And this author concluded in their Cell paper that it is beneficial to add the M protein to the S component vaccine, which I think nobody else has done that. And we will see if that brings any advantage to our vaccine compared to the other one. In addition, that we are delivering a VLP by a live virus vector. So our third construct is just using RBD, and we are delivering RBD with a non-COVID-19 matrix protein in terms of VLP. And the reason people do the RBD is that to get away from the S protein with SARS-1, the C terminal of S protein was shown to be involved in immunopathology. But there is again enthusiastic with the RBD is also dampened by some publication showing there have been a couple of monoclonal antibody to RBD of that has been binding to the RBD and kind of keeping the structure of the spike protein in an up position and kind of enhancing the infection of the susceptible cells. And that's in vitro, and that's with monoclonal antibody. If the polyclonal response to RBD is doing the same thing, it is not known at this time. So that would be our third construct. So this just shows on the left that this is our 6 kb. This is a large fragment that we have inserted covering the spike M and envelope protein of Wuhan strain. In the middle, you see the spike protein, expected size 150 kilodalton. And on the right side, we have stained the live virus with anti-spike monoclonal antibody, showing the conformation of the spike is a native conformation. So we developed a hamster challenge model it is performed at UTMB with our collaborator. And they, as you see here, they did the animal, the challenge with 10 to the 5 PFU of SARS-CoV-2 intranasally. As you see, the control do not show any weight loss, whereas the infected animal, they lose weight. And on the right, you see that the virus can recover from the lung and nasal turbinate from these animals on day 2 and day 4. So this is the animal challenge model that we are going to use, and we have started this experiment. Animals are vaccinated by intramuscular 1 dose or 2 doses, and they will be challenged intranasally, and they will be looking at binding antibody, neutralizing antibody, and also protection, and also we will look into the lung for any sign of immunopathology, which I think is giving us very important information to move forward. So summarize, we have constructed and designed 3 candidates, and we are going, the next step after the challenge model in hamster, we are going to the H2 and non-human primate. And we have talked to FDA, and they have agreed that we don't need any toxicology for COVID-19 because we have an excellent safety record of our vaccine in human. So just summarizing, we have shown 12 indication, and we have validated platform. And for HIV, we have completed phase 2A. We are ready for pivotal trial. And we show proof of concept in 6 indication: HIV, Zika, Ebola, Marburg, Sudan, and Lassa. And we have a huge collaboration, which helped us to move this program forward very quickly. And so far, we have invested more than $100 million in the platform. And just acknowledging our GeoVax colleagues who did the work, and also UTMB and USAMRIID doing the Lassa, and UTMB did the Sudan and Marburg, and Ebola was done at Rocky Mountain NIH lab, and CDC work on our Zika vaccine, and we also collaboration with malaria with Leidos, and HVTN has been running our HIV clinical trial. Thank you very much.
Thank you very much, Dr. Girahu. Excellent talk. We really appreciate it, and I can tell you that it's very pleasing to see that the MVA VLP technology that's been developed at GeoVax has been well validated against a number of different other viruses, and it's good to know, it's comforting to know that this may also be utilized very effectively for COVID-19. I can tell you that I've gotten a lot of questions for you in the chat box. The top #1 question, I think, was addressed by your next to last slide or so, which was the status of your animal model studies. And so you had mentioned that you're initiating, I think, the hamster studies. Do you have any preliminary results at all with your vaccine?
No, the animal were inoculated with the first dose last week. So we will be waiting for the group with the second dose. So we'll have the data for single dose efficacy and also 2 doses, and we will get the data for neutralizing response, and also we get the data for protection. Although FDA has given everybody to go ahead without having any protection data to phase 1, and we are going to show protection before we go into phase 1 because we have time to do that.
Right. Very exciting. We look forward to seeing those results. And let me just ask you a general question, because it's come up throughout the webinar today, and that is, there's a lot of concern about the long-term vaccinogenic protection or protection against COVID-19 given what everybody suspects is the ability of the antigens to have variation over time, or the mutations of the virus. So can you comment on what you think the challenges are here, or maybe there's going to be a lot of success in a single vaccine actually protecting us long term?
Yeah, so I think that the COVID-19 is not like flu vaccine, it's not segmented genome, and it's not going to do antigenic shift and drift, which we would need a seasonal flu every day and need to change vaccine. There are some mutation has been shown about, I think, maybe 20 nucleotide changes in some report and so on, and maybe changing some amino acid, but the spike protein is a huge protein, is a big protein. So even if you just use the spike protein, you produce neutralizing antibody against many epitopes. So if one epitope is changed, you still have redundant neutralizing antibody in polyclonal response that should take care of the virus in terms of neutralizing the virus. But also it's important to have a T-cell response because you want to get rid of the infected cells. And that's why the T-cell response, CD8 positive T-cell response is important, because in some patients who were infected with COVID-19, they were protected without showing any neutralizing response. So there must be a T-cell response. So what we are focusing here, we induce antibody response and also T-cell response. And our T-cell response is not only limited to the S protein, but we also have T-cell response that I show you in my slide that is also directed to the M protein and also maybe E protein. So we have the whole three important structural proteins: spike glycoprotein, M glycoprotein, and pentamer E protein, which cover lots of T-cell epitope, which would be redundant. And we hope that we have a broad protection than other vaccine, which might be just solely based on S protein or maybe limited just to RBD.
Yeah, that question really hits home, I think, for GeoVax, and I think it shows the true value of your technology to hopefully inoculate and vaccinate long term, because you're looking at multiple different antigens at the same time using your technology, right?
Exactly, and with our HIV clinical trial in human, we have shown the duration of the antibody with our platform is much longer than the other pox viruses. So when we compare our clinical data, our HIV with RV144, which was the only clinical trial which showed 31 efficacy, the level of anti-GP120 in our vaccine after 1 year was similar to that trial after 6 months. And the antibody dropped in that trial after 1 year, and that's why the efficacy was lower, whereas our antibody remained high even after 1 year. But we also induced T-cell response, which was superior to that RV144. So with our clinical trial in human, we know we have durable antibody and also T-cell response.
Great. It's very exciting, Dr. Gerhu. We appreciate that. And we've got a number of other questions, but I believe we're just about out of time. So I'm going to ask our host, Elizabeth and CHI, to email those to you. But we're very excited about what's occurring at GeoVax, and we wish you and Mary and others there the best of luck.
Thank you so much.
And so with that, I think it's time to conclude this online webinar. And I want to start by thanking the speakers, Dr. Hotez, Dr. Gerhu, and my colleague, Jiawei Yang, for the excellent presentations. I also would like to thank Elizabeth Lamb at CHI and CHI specifically for hosting this conference and allowing Sino Biological to be the eminent sponsor of this conference. And finally, I want to thank all the registrants and all the participants who listened in, and thank you for your questions as well. We really do appreciate your enthusiasm and the very strong turnout. And then finally, my colleague, Jen Liu at Sino Biological, for setting all this up. Appreciate your help as well from a marketing perspective. This was a big success for us. So thanks to all. And I guess with that, we'll now adjourn.